Laksh's uncle is a great gardener and this is his orchard. Here he grows mangoes, roses, grapes and cashews. Laksh thinks his uncle's fruits are quite delicious, so he decided to grow the same plants in his garden. But he wondered now if he could grow these plants quickly without compromising the taste of the fruit. His uncle suggested he should grow the plants artificially by using parts of his plants and trees. The process of growing plants artificially is called artificial propagation, but it is not as simple as it sounds. Artificial propagation is a type of vegetative reproduction in which breeding or growing plants is done using parts of the parent plants like stems, roots and leaves. So for roses, Laksh's uncle cut a part of his own rose plant with a bud and told him to fix it into the soil in his garden. This method is called cutting. In this method, a new stem grows from the branch fixed into the soil. This new stem grows into another rose plant. And guess what? Because he used the part of his uncle's rose plant, Laksh's plant will bear roses just as pretty as his uncle's. Next, for those delicious mangoes, Laksh's uncle suggested he should cut small shoots or buds from his plant stem and attach the cut portion to the plant in his garden. This process is known as grafting. The cut portion of the parent plant is called scion and the fixed plant with its roots is called stalk. Even for grapes, his uncle gave him one of his own plants. Laksh wasn't sure what to do with just one plant, so his uncle told him to bend one of its lower branches to the ground and cover it with soil. This process, known as mound layering, leads to the development of new roots. Soon, Laksh will have a whole row of grapevines and a fresh bunch of grapes to taste. This method is similar to cutting, except that here the branch is left attached to the plant. Lastly, for cashews, Laksh's uncle suggested he should remove the bark of a branch from one of his plants, then cover the area with moss or cotton bandage and water it daily. This method is called aerial layering. Laksh should have his cashew plants ready in a few months' time from now. Laksh has now done everything his uncle has asked him to do and is quite excited to see the fruits of his uncle's advice.
A number of artificial methods are used to propagate plants in agriculture and horticulture. Some of these methods are layering, cutting, grafting and tissue culture. Let us discuss about these methods. Layering. In this method, a lower branch of a plant is bent down close to the ground and covered with moist earth in such a way that its growing tip remains above the soil surface. After a few days, the covered portion of the stem usually produces adventitious roots. The rooted branch is then cut and grown as an independent plant. This method is used to propagate plants such as jasmine, and it occurs naturally in black raspberries. Cutting. For cutting, we have to select a healthy young branch with a few nodes and internodes. Then cut off the branch and remove most of its leaves. Now the cut end is stuck into some good moist soil. Look at the cutting after some days. We can see that it develops roots and grows into a new plant. This method is used to propagate plants like rose, sugarcane and pineapple. Grafting. The art of joining parts of two plants in such a way that they grow as one plant is called grafting. For doing grafting, select a plant and then detach a twig or bud from it. This twig is called scion. The scion is then kept over the cut stem of another plant. This is called stalk, and it has an extensive root system under the soil. The two cut surfaces are bound together and the joint is covered with wax to prevent evaporation and also to stop infection. The tissues of the stalk and scion join together to form one plant, with the stalk supplying all the essential nutrients to the scion. This method helps to combine desirable features of two plants. For example, a high-yielding variety of a fruit is grafted to another of the same species known for its toughness. Then we get a tough, high-yielding variety. Mango, rubber, apple, pear, citrus and guava are propagated through this method. Tissue culture. It is a modern method of vegetative propagation. Let us understand the steps in this process. For tissue culture, first cut a piece of tissue from a desired plant and then keep it in a nutrient medium under controlled conditions. This tissue grows into a mass of cells called callus. Later on, callus formed is transferred in a different nutrient medium, which makes them develop into new plantlets. These plantlets are now grown in pots of soil. Tissue culture is used in the propagation of orchids, chrysanthemum, etc.